Hello and welcome to The Explainer. Today, we're diving into the incredible journey of biology. But this isn't just a story about discoveries, it's a story about the tools that made them possible. We're going to see how science went from what we could literally just see with our own eyes to what we can now build with software. Let's get started. I mean, seriously, can you? It's almost impossible, right? Technology is just woven into every single part of our lives. And it's the exact same for science. Technology has been the engine behind this massive explosion of biological knowledge. So how did we go from just observing nature to actually being able to change it? Well, it all started with the most basic tool of all. You see, before we can even begin to understand the world, we have to be able to measure it. But in science, not all measurements are created equal. You have to speak the language of measurement. And that language is really built on two crucial ideas: accuracy and precision. Okay, first up, accuracy. The easiest way to think about this is just hitting the bullseye. It's all about how close your measurement is to the real, actual, true value. You know, if a baker's scale says a 1-pound loaf of bread weighs exactly 1 pound, well, that scale is accurate. It's simply about being correct. And then we've got precision. Now, this is different. This isn't about being correct. It's about being consistent. So let's say you take the same measurement 3 times and you get the exact same result every single time. Your measurement is super precise. But here's the catch. That result could still be totally wrong. Precision is all about repeatability. So how does this actually work in practice? Okay, picture this. You're tossing horseshoes. If all of your tosses land in this tight little group way off to the side of the stake, well, that's high precision but low accuracy. You're very consistent, but you're consistently missing the mark. Now, what if your tosses are kind of all over the place, but their average position is right on the stake? That's high accuracy but low precision. You're sort of right on average, but each individual attempt is, well, it's a bit wild. And this, right here, is the bullseye. This is what we're always aiming for in any scientific measurement, to be both accurate and precise. In our little horseshoe game, this means landing every single toss in a tight little cluster, right around the stake. That is the gold standard. It's getting the right answer over and over and over again. Okay, so once we mastered measurement, science was ready for its first huge technological leap. I mean, think about it. Until the 1600s, nobody even knew cells existed. And then boom, the microscope was invented, and suddenly this entire universe that was hiding in plain sight was revealed. Now, this tech didn't just pop up overnight. Those first microscopes back in the 1600s, they gave us these blurry little glimpses into a brand new world. It actually took 200 years for better lenses to come along and really sharpen that view. But the true game changer? That came in the 1930s when scientists figured out how to use electrons instead of light. And that changed everything. What's so fascinating here is the trade-off you have to make. A light microscope, you know, the kind you probably used in high school, it can magnify things up to about 1,500 times, and this is key. It lets us look at living things. But an electron microscope, whoa. It uses beams of electrons to magnify things up to 1,000,000 times. That's powerful enough to see individual protein molecules. But here's the catch. The specimen can't be alive. It has to be in a total vacuum to work. And it gets even cooler because even within the world of electron microscopes, there are 2 main types that give us completely different ways of seeing the world: the scanning electron microscope, or SEM, and the transmission electron microscope, or TEM. So what's the difference between them? The scanning electron microscope, the SEM, is all about the surface. It scans a specimen with a beam of electrons, and then a computer uses that data to build an absolutely beautiful 3-dimensional image. This is how we get those incredible, super detailed pictures of things like an ant's face or the surface of a grain of pollen. The transmission electron microscope, or TEM, does something totally different. Instead of scanning the surface, it shoots electrons through an incredibly thin slice of a specimen. What you get is a 2-dimensional picture that shows you all the internal structures. So while an SEM shows you the outside of a cell, a TEM lets you peek inside and see what's going on. So the microscope, it opened up this incredible, invisible world. But
what about the systems that are way too big, or too dangerous, or just too complex to ever fit on a slide? For that, biologists had to turn to another revolutionary tool, the computer.
And this is so important. Computer models let scientists run experiments that would be completely impossible otherwise. We're not just talking about convenience here. This fundamentally changes the scale of the questions we can even ask. You can't just release a new virus into the wild to see how a pandemic might spread, right? And you can't possibly track every single animal in the Amazon rainforest. But with a computer, you can simulate these massive complex systems and watch it all play out.
Let's take epidemiology, the study of how diseases spread. A researcher can plug in all the variables. How contagious is this thing? How long before you show symptoms? What are the chances of becoming immune? Then the computer just runs thousands and thousands of virtual interactions. And this doesn't just give you a prediction, it can reveal these hidden patterns and vulnerabilities in a system that you would never see in the real world. It's an unbelievably powerful tool for protecting public health. And this brings us to the ultimate frontier. This is where all these tools, precise measurement, powerful imaging, and complex computing all come together. I'm talking about molecular genetics, the study of the very code of life itself. At the very heart of all of this is the gene. It's pretty simple, really. A gene is just a segment of DNA that holds a specific piece of genetic information. For decades, we knew these things existed, but to actually read them, to understand them at a molecular level, that took a full-blown technological revolution. And this is where things get really, really wild. Molecular genetics gave us the power to read and even tweak individual genes. That then scaled up into genomics, the ability to compare the entire genetic library, the whole shebang of different species. But I mean, how do you compare billions of lines of code? Well, that's where data mining comes in. We have this powerful software that just churns through these massive genetic databases, finding patterns that can tell us everything from our evolutionary history to our personal risk for certain diseases. Just stop and think about that leap for a second. In just a few decades, we've gone from barely being able to read the genetic code to actively rewriting it. This is the science that's behind everything from crops that can survive a drought to the incredible potential of gene therapies that could one day correct inherited disorders. It's a direct result of putting all the tools we've talked about today together. And that brings us to the end of our explainer. From a simple ruler to a supercomputer, technology has constantly been redefining what's even possible in biology. So the real question isn't just about what we've already discovered, but what will the next generation of tools allow us to find? What is the world of biology going to look like in another 20 years? It's pretty amazing to think about. Thanks for joining me.